longer than some of you have been alive. So, Steve, all you.
Okay, so first of all, don't hesitate to interrupt me at any time with questions. Some of the material may be very new to you. It may not necessarily be important that you understand some of the subtleties, but I'm going to try to present a problem that we've been working on. I've been working with the Institute for Systems Biology, which is a local organization, and EMBL-EBI is a European biolab, and also with Uninet, which is a cloud computing company in Norway. We'll ignore the abstract. This is a war story. We have a big problem. We're going to talk about how we parallelized it, what kinds of problems I encountered, and the tools that I wrote to help me deal with real-world problems. So it's a real problem, and it actually predates, I think, even the existence of Spark. So how do I get into this? Well, a few years ago, I wrote a Hadoop proteomic search program. I'll tell you a lot about what proteomic search is later on. But I wrote this program in Hadoop. It was good. It ran on large data sets, and we published a paper on it. And then about a year ago, I got an email from Gurvinder Singh, who is at Uninet, a Norwegian company, which says, "Hey, how would you like to do this in Spark?" And I said, "Sure." Because he had been working with their cancer lab, and they were interested in the problem. And they had a Spark cluster, and they needed a problem to work on. So that's how I got into this. So let's think about the problem. You realize in biology that we start with DNA, which encrypts and encodes information. And what the information that DNA encodes is the structure of proteins. Now, a protein is a string of amino acids. There are 20 amino acids in the body, and so you can represent the structure of a protein as a string with a 20-letter alphabet representing the 20 amino acids in that protein. Because we can read the DNA, we have a pretty good idea of what proteins we're likely to find in the body. So, we also have some interesting tools. One of the tools is several enzymes which will break up proteins in known and interesting ways. So, we're going to use trypsin a lot, and trypsin is an enzyme which will break a protein after an arginine or a lysine, which are two kinds of amino acids. So if I have something like this, you note there's an arginine here, but it's followed by a proline, so it won't be broken there. And a lysine here, and when I break this up, I'm going to get 4 fragments. If I just dip this in trypsin, it will break things up. That represents digestion. And so, they've developed about 10 years ago a technique called tandem mass spectrometry. And this is a sophisticated technique to look at a sample which contains a number of unknown proteins and figure out what's there. So what you do is you take a sample with a bunch of proteins, you digest it with trypsin, breaking it up into a lot of fragments. You run them through a column which just separates them out so they come out one at a time, and then you stick them in a mass spectrometer. And a mass spectrometer is an instrument which, in ways that I will describe in some later slides, is going to yield a spectrum. And once you have this spectrum, you compare it against all possible spectra from all possible fragments of all proteins that we know about in the human body. And that turns out to be a big computer problem. Okay, back to the mass spectrometer. So our our peptide fragment comes in. We hit it with something which ionizes it, because mass specs only deal with ionized stuff. And then we measure its mass to charge ratio, essentially its mass. After that, we hit it with another thing which breaks up the peptide bonds. So you've got a sequence of peptides and you break up the bonds into a series of fragments. And then the next stage measures the mass to charge ratio of all of these fragments, and it gives you a spectrum, which would look something like this. So we have a series of peaks representing the fragments that we see in the mass spectrometer. So in reality, if you go in the lab, you will take a sample, digest it, run it through your mass spectrometer, and you will get tens to hundreds of thousands of these spectra as you analyze the sample coming through your system. So you get a lot of spectra, and now you have to figure out what they mean. So, where do you start? Well, you go to a well-known site, and they have databases of proteins. You name an organism, human, mouse, yeast, E. coli, all known organisms, and you can download a file with all of the proteins that are there. These are frequently annotated. There are notes because this is biology. Things are never simple in biology. There are rules that are followed most of the time, and then the rules get broken. But in any case, the kinds of numbers you're dealing with, the human protein database is about 20,000 proteins, about 20 million amino acids. In general, in search, you are going to roughly double the size of the database, because for every real protein, you add a protein which isn't in the database, because if you find that, that's telling you that this is just a random find. It's not something that you should look for. These are called decoys, and it roughly doubles your database. And if we deal with multi-organism databases, they can run in the 500 meg range. So the moral is, these are pretty big to start with. I should turn mine off too.
All right, everybody gets to turn off their phones for just a second.
So what do you get when you say, give me the proteins? You get a file which looks like this. It's a FASTA format. Essentially, the first line is a comment, which is an identifier for the protein, and then these are all the amino acids, and then anything that starts with a greater than sign is another comment, and this goes on for many megabytes. There isn't anybody in this room who couldn't write a parser for this file in their sleep, and most biologists could write a parser for a FASTA file. Writing a Hadoop input format, which is what you'd like to use with Spark, is only slightly more challenging. So, you start with the protein database, and then you do what's called in silico digestion. And in silico digestion means you do exactly what the enzyme would have done with the protein. So, you're going to take the protein and break it up like this protein. And again, we break it up with trypsin. I've shown you this slide before, but now you're doing this in the computer to come up with all of the fragments that you expect to see. So you start with a list of fragments, and now you go back and you add chemical modifications. It turns out that there are chemical groups that can bind to amino acids. Sometimes they're there, sometimes they're not. And for every modification you have to generate the fragment with or without the modification. So this right here represents a methionine with a hydroxyl group which has a mass of almost 16 Daltons added onto it. These are serines with various modifications added, and modifications increase your search space a lot. Typically, people will ask for somewhere between 4 and 8 modifications, and that number is probably going up. So we did digestion, and, well, almost. Sometimes the trypsin doesn't cleave right, so you tend to stick a couple of fragments together and treat that as a separate fragments. Sometimes breaks occur in other places. You may worry about that, and there are chemical modifications. Sometimes there are isotopes involved which change the masses that you're looking for. And of course, they may or may not be present, roughly doubling your search space. So all of these changes push the number of scored peptides into the tens of millions or even up into the billions. Oh, fragments. So anyway, you have a peptide like this. And this is 10 amino acids. And it can break at a series of different bonds. So it can break at the first bond, the second bond, the third bond, yielding 2 sides. The left side is a B ion and the right side is a Y ion. And there will be 20 of these for each possible bond, and you can compute all of their masses. Given that, you can say, if I had the peptide shown in the previous slide, I would expect a spectrum which looks like the upper colored spectrum. And this is something that a computer can readily do, and now we have to compare the measured spectrum with the upper theoretical spectrum. Usually there are a lot more peaks in the measured spectrum, and all you do is you run through the 2 spectra, and if the peaks are close enough, you say there's a match. This is called cross-correlation. So the lines, the peaks that I have in red, and you might not be able to see the text, are ones that line up and all of the others don't. So based on cross-correlation, you get a score. This spectrum matches this peptide. So you take a weighted sum of all of the peaks, and this gives you a score. You score your spectrum against all of the peptides, and the best scoring peptide is assigned to that spectrum. Typically, and this is where we're dealing with biology again, you get a good identification on maybe 20% of your spectrum. The rest of it is just junk. It just works out that way because there's a lot of interesting chemistry that goes on, and we're not modeling the real world quite as well as it turns out to be true. So you're doing a big search. You're going to get some results which are interesting. 20% of your spectra is more than enough to identify the proteins, but there's still a lot of noise in all of this. And why is this a big data problem? Because we've got a lot of spectra multiplied by a lot of proteins. And at this point, we're going to kick off a Spark job on our cluster and go back to the presentation, if I can find it. That wasn't it. That's it. Okay. So what I just did is, using a PuTTY terminal to talk to the cluster, which happens to be in Norway, I sent the following command: a Spark submit, asked the cluster to run a job. I gave it the main class. I gave it the jar with all of my code. And I gave it 2 files, one of which is a file which a standard program in biology, we'll talk about politics in a second, would handle, and just describes the biology of what we're doing. And the second file, this Spark cluster UPG, describes things that are specific to that Spark cluster, which is where on HDFS do I find the data, what are the parameters of the Spark cluster I intend to set up, and a whole lot of knobs that I would use for tuning the system because I found that they were absolutely necessary. Okay, politics. So I have to sell this to biologists when I get done. And this puts some constraints on the system. The biologist said very specifically, here are some programs that we trust. They're in C++, they're written by biologists. Anybody who's ever seen code written by a biologist knows what I'm talking about. So I ended up taking one of the better written programs and translating it into Java. I had to accept some constraints in terms of the way it handled memory and how much memory it was going to use, because if I didn't use the biologist's algorithm, they weren't going to believe the results. So, anyway, this is a big problem. 50,000 spectra, and we're now analyzing up to 400,000 spectra by a couple of billion peptide fragments turns out to be an extremely large number. The good news is you don't have to do all of those pairs. All you have to do is the ones who you know the mass of the theoretical peptide fragment, you know the mass of that you measured for the spectrum, and they have to be close enough to look at this. So they have to be within the same bin, which in a high precision instrument ends up being about 0.3 Daltons, which means you're scoring a lot less than that very large number we came up with. It's still a lot of scoring, but you're doing a lot less work and putting things together in these bins, all spectra with a particular mass, with all peptides of that mass, and scoring them together makes the problem tractable. So, we put things in bins. In general, the rule was that we put a peptide in one bin and a spectra, in order to cover a range of masses, a spectrum was replicated in multiple bins, all the bins that were close. And then if a bin got too big, we simply subdivided it. So we end up holding N spectra and K peptides, and in the current problem there are tens of thousands of these bins. And now we get into war stories. As if your bin gets too big, you run out of memory and the whole thing fails. One of the rules in Spark is every task you give it has to succeed. And if you have 10,000 tasks and 10 of them fail, you're going to fail. So figuring out which were the 10 that have failed and how to fix them turns out to be a critical problem. So anyway, what did I do in Spark? I read a database of proteins. I ran a Spark routine to digest them. That takes one protein, breaks it up into fragments, and gives me peptide fragments as an RDD. I added modifications. Both of these are flat maps. That is, one will go to many. And finally, I put these into bins. Similarly, I read the spectra. I normalized them in interesting ways that the biologists want to see. And then I put the spectra into bins. That is also a flat map because spectra are put in multiple bins, as are peptides sometimes. Then we co-group the bins, which is to say, we do a co-group which puts all the spectra and all the peptides together in one task that we're going to analyze in one function. After that, we run a score, every spectrum against every peptide. And once we're done, the spectra have unique IDs. So we've scored a spectrum in a number of different bins. We sort all of the best scores we got in the bin. or all of the scores we got in the bin, we sort them, choose the best one, and that's the answer. And finally, we report the best fits. That's basically the program, which, let's switch off and go over here and see how it's running. Let's see here. Come on. Okay, so we're now off and running on the cluster, and we have a series of jobs that are up and running. At the moment, we're still reading these spectra in. We haven't gotten to actual scoring yet, but we will. This job will take about 15 minutes to complete. So, back to the presentation, debugging. We started with an unfamiliar problem running on a relatively unfamiliar platform. Not a lot of people in this room have massive experience solving many problems in Spark, and I certainly didn't have any of this. So I was left with lots of questions, like which operations are taking the most time? Where is my time being spent? How many times is each function being called? Have I balanced performance? Have I balanced the work across the machines of the cluster, or is one machine doing all of the work? And when a small number of cases are failing, how do you instrument the system to find the cases that are failing? So, did it work the first time? Hell no. Things never work the first time. Did it work the second time? Well, no, it didn't work that. After much work, after we got it to stop crashing and to run a fairly trivial program, we ran a sample that we really cared about, and it took 30 hours real time to run on the cluster, which incidentally the thing would have run in about 4 hours on a single machine. So we weren't doing very well. And at that, yes?
On a single machine on Spark? Oh,
No, on a single machine using
C++?
C++. The 15-node Spark cluster runs about 3 times faster than a reasonable single machine because the C++ gets to do some real optimization and in ways that Spark can't do it. So we're going to have to throw more nodes at it to really start getting a lot better than the single machine. At that point, I started looking for tools to figure out what was going on. The biggest thing I found had to do with a bug in my Hadoop input format, which was causing data to be read multiple times. It largely dealt with the fact that it hadn't been tested very well on really large files, and the format I was used to was a little bit different than the one I was being sent. One of the things that I discovered, and now we get to see some code, Spark accumulators. Hadoop has something called a counter, which is a number that you can increment across the system, and when you get done, it will give you a total of all of the increments made across all machines in your cluster. Spark has a concept much more powerful than a counter called an accumulator. And what accumulators can do is take any two objects which can tell you how to add them together and keep a running object representing adding everything that was done in all of the nodes in the cluster. So for example, let's see if we can go full screen here and get rid of that. Let's assume we're back at the word count problem, And we just want to know all of the words that have ever been encountered in a large set of documents. Well, you can keep track of words by just having a set of strings. So all you have to do to make an accumulator for the words is to say, I'm going to create a set accumulator which implements a cum param of a set of strings. And the really tricky method is to say if I have two sets of strings, I add them together by creating a new set, adding all from the second set and returning the new set. There's an add in place which will do essentially the same thing. And 0, if I pass in a value I call 0, what do? I'll return it. That's it. And now to use this, in the middle of your word count, what you can say is, okay, I take my line, I break it up into words. Now I take the words and I add them all. I create a new hash set with all the words and I add them to this accumulator. Done. We now have all of the words that we have ever encountered anywhere in the system. Obviously, I am not interested in word count. What I am interested in are things that matter to me, which is to say, what I've done is for the functions that I use, and I'm using the Java version of Spark, instead of implementing the functions like flat map function to pair function, I implement a class which has already implemented these, abstract logging function, abstract logging pair map, and instead of implementing a call method, I implement a do call method. A base class wraps all of this with instrumentation to count how many times it was called, how long it took, and one other thing, which is how many times did it execute on each MAC address, which tells me what machines is it running on. So then my implementation looks like this. This is an ordinary function 2, but we call it an extend abstract logging function 2, implement a do call method, do whatever I'm doing. In this case, I'm choosing the spectral match with the best hyperscore, which turns out to be what you would do to get the best score from once you have all the scores for a spectra, you want to choose the best one by just returning whichever one was best. And this is instrument. What does it give me back? It says, I have this function. I called it 69 million times. It took a total of 29 seconds. It ran on 15 machines, and the variance is the difference between essentially a statistical calculation on how well spread it is. 0 is good, 1 is bad. So 0.5 is pretty good. So let's see how our job is doing. Okay, so we're nicely running this job, and in the middle of a job, as things succeed, the information from these accumulators becomes available in a report. It might take a while because I'm running a lot of stages here, but here I have a bunch of jobs that are running. It's still downloading stuff, so I should go back to the presentation and wait for this page to, oh, there it is, okay. So these are my accumulators. So I don't know how much you can read this, but what I'm seeing is the critical time is I have scored this function which scores a single bin that has been called 70,000 times. It's taken 293 minutes, which is order of what? How many hours? 6 hours? Right. 6 CPU hours on 15 machines, And it still has a pretty high variance, probably because not all of the machines have started running this function yet. We've called this split map polypeptides. This is a function which essentially is run for every polypeptide. So we can see we've got about 600 million peptides that we're looking at here, and it took 10 minutes, which is not going to be a big time compared to the 4 hours. One of the things I discovered rapidly with this technique is the scoring function took all the time. You could do anything else you wanted to this job, and you wouldn't affect it unless you could make that scoring function faster. This caused much experimentation in partitioning and counting and trying to distribute scoring better because it wasn't touching the performance of the system. Okay, back to the presentation. That wasn't it. Okay, good. Yeah, that says go back to your running job. Let's see how it's doing. So, what did we do to improve the performance? I fixed some bugs. I discovered that most of the time has been spent in scoring. We ended up using a Parquet database to store the results of digestion, because we're tending to do digestion on the same file every time. That really isn't necessary. You can store it in a database and retrieve it. That saves some time. I discovered that repartition is not a very expensive operation. I discovered that counts are not a very expensive operation. One of the things we did is we got this running. I'm currently running a sample of 10,000 spectra. We got that running 5-6 months ago. And it wasn't that long ago. But my colleague in Norway who is into big computing says, can you do 1 million? And that took a lot of tuning. So far, I can do 400,000 without hitting memory errors. But one of the things that I discovered in scaling up is that I wanted to increase the number of partitions that I was doing, that small numbers of partitions were killing me on memory. So we started off with a default parallelism of 200, we went to 2,000. On my larger problems, the number of partitions I'm using is getting closer to 10 to 30,000, because it's just failing if I start to cut that number. The smart partition said I started to realize that because I know the structure and because I can count the number of spectra and the number of peptides that I'm going to score in a later stage, I can make decisions about which bin goes into which partition so that the bigger bins get their own partition and the smaller bins, you can put several of them in the same partition. And that made a difference.
Question.
Yes?
What do you mean by Parquet database?
It is
Parquet
a...
files?
What? Yes. Well, it's a system...
The Parquet file format.
Right, Parquet file format, yes.
Okay, you don't mean
Yeah.
a database.
Okay.
Can you talk a little bit more about partitioning?
Yes.
I'm not familiar enough with partitioning. Can you talk about the background?
When you process an RDD, one of the things that you can give it, normally what it does is if you have a specific number of partitions and there's a default number that is set in the system. When you run an RDD, it will break them up. If it's a pair, it will look at the key, it will compute a hash for the key, and if you've got 200 partitions, it will hash it into 200. But one of the arguments in the Spark calls allows you to supply your own partitioner. And a custom partitioner can tell you how many partitions you're going to have and how a particular key is going to be assigned to partitions. So you can write your own custom partitioner, and what I ended up doing is I looked at all of my bins and I created, based on the counts, I created a hash table which says if it's in this bin, it goes in this partition. I generated a partitioner and I passed it in and I said, that's what you're going to ask for. So it's taking fine control over the way the thing is run.
So you've gotten to 400,000, trying to get to 1 million. What's happening to prevent you from getting there?
A small number of samples are running out of memory. And I'm still trying to figure out exactly what are the characteristics of the ones that fail. One of the problems that you run into is that accumulators are absolutely great and you can pass back a ton of data, but you only get it when things succeed. So it's the hard problem of, okay, I did it 30,000 times and 100 of them failed, but I want to look at those 100 rather than the ones that did report.
Now, if you have all 1 million and you broke them into, say, 3 batches of 300,000
That would work.
So each one independently will
Yes,
complete? Okay.
I believe it will, yes.
Interesting.
It's just a question of bulk.
And this is still on a 15-node cluster?
Still on a 15-node cluster. I'm twisting the arms of the people in Norway to raise the number of nodes to, say, 100. See if that helps things. I'm not sure it will.
Yeah, I wonder if theoretically, as long as you break it into enough partitions, a small cluster should be able to handle a high number of partitions as well.
Just
as
takes
big
more
ones,
time,
but,
yes.
But yeah, it's an interesting challenge that you've hit there. You have me curious now.
Okay. I'll take all the help I can get. So, okay, smart partitioning. I talked about smart partitioning. You can count the number of items you are going to process in a partition in advance, because that's a cheap step. And then you can base the bin size, you can base the partitioning on trying to put the same number of operations in each partition based on what you know. That actually kicked me from maybe being able to do 50,000 to 400,000 on this problem. So these are older numbers. I've gotten things to run better and with larger samples, but roughly we get about a 3-to-1 improvement over a single machine. We think we can do better on a larger cluster. That is, simply adding more nodes should increase the performance of this. We've also done more scoring, but this is 605 million peptides is about the right number, and I think the job is done and we can actually look at some reports on what we scored. Yeah, the job finished.
Question.
Yes.
How does that compare to your Hadoop implementation?
That's a good question, because I don't know that I've run the same thing on the Hadoop implementation and the Spark implementation.
Steve?
Yeah.
Repeat the question. Could you repeat the question?
Sure. How does that compare with the Hadoop implementation? And I'll be honest and say I haven't run the same problem on the two systems. We're using a different lab's data, but I probably should do that. My instinct says that they're probably also, I was running on a different cluster for the Hadoop stuff. My instinct suggests that the numbers are probably fairly comparable. The Hadoop stuff is forced to write to memory at different stages. We use fewer stages in the Hadoop cluster than we used in the Spark implementation. But the critical stage where the scoring was done was pretty similar. The other thing is the algorithms being used are somewhat different. The problem with the Hadoop implementation is it used an algorithm that worked very well for low resolution data, which was largely what you found at the time I wrote it. And now almost all the data is high resolution, and I didn't trust the algorithm that the Hadoop version was using. That is the Java code that did the dirty work of scoring. I could put this back in the Hadoop implementation and figure it out. So when we get done, I get a series of reports. These are, we scored 200 million peptides and 290,000 spectra, and these are the times that functions ran. As you say, many of them ran in the millisecond range, and the one score spectrum with cogroup without hash is the one where the total time was almost, what would that be, 6.5 hours. That's where all the time was being spent. The samples that I'm running now with 400,000 would run for closer to an hour and a half and would be burning hundreds of hours of CPU time.
Roughly how much RAM are you using? Sorry.
I'm assigning each slave up to 14 gigs. And we played with that number, and we kept trying to raise it. And I'm not sure whether it's a good number, but if you drop it much, things start running out of memory quickly. And I don't think I get to raise it on this cluster.
So you have 50 nodes, and your each slave has 14 gigs. And
Yes.
then your executors, you're talking about executors?
I'm talking about executors, yes.
And there is only one executor per node?
What?
How many executors are running per node?
I'm not certain.
This is running on Databricks Cloud. Because you were using the UI, right? It looks like it was a.
They're running Mesos. And if this is a Databricks, somebody from Databricks, tell me whether it's their UI or not.
It is.
It is, okay.
Well, I'm not from Databricks, but.
Okay,
we
it's
use
their
it.
UI.
It looks like their UI.
That's just the Spark UI.
That's- okay, that's-
not Databricks Cloud.
Okay.
That's not the Databricks.
Oh, okay.
Okay.
UI. That's the Spark UI where you can troubleshoot stuff.
Okay.
Yes. So this thing has finished, so if I were to refresh the screen, it would tell you that the job isn't running anymore. So let's see if I can find my presentation again. Oh, come on. There you are. Okay, so in summary, we have a large problem involving a large number of spectra scored against an even large number of peptides. In the future, we believe that the complexity will increase. We're going to get more spectra because instruments get better. We're going to get more peptides because biologists are going to want to add more chemical modifications and isotopes looking at peptides. Spark is a parallel execution. We can do the search in Spark. We can do better than single nodes. And as we scale the size of the cluster, we expect performance to improve even more. Okay, thank you. Let me take questions. Questions? Yeah.
What version of Spark are you using?
What?
What version?
Of Spark?
Yeah.
14.
Maybe we should try this.
Oh, nice.
Any questions? Anybody?
Question.
For the purposes of the video, could you repeat the question?
Okay. The question was, what version of Spark are you using? And I'm using 1.4. Other questions? Yeah.
Can you parallelize individual components in Spark? Like say, figure out how many kinds, like for Hadoop we use numbers and reducers. Can you, like based upon each of the components, you show different timelines, some took 6 hours, some took much shorter time. Is it possible to, wherever the time is more, is it possible to rescale it to performance?
In
Does it
each,
provide it?
in each of the Spark function calls, every time you operate it on an RDD, you have the option to pass in a partitioner, which in effect divides the work. So in that sense, you have control of the division at an individual function level if you choose to use it. That might not always be the best decision because Spark might be able to perform some of the more trivial operations like filters locally in memory, and it might make sense not to move things around. But what I found in this program is this problem is CPU limited, and almost anything else you're going to do isn't going to touch performance.
The logging he was doing was making that call across all of the workers in the cluster. So the time that he was reporting there was the aggregation of all of those jobs that were already parallelized across the cluster. So the only way to really improve the real time for it would be add more nodes to the cluster.
Other quick, yeah?
Yeah, what is the workflow of the operator expected to be like? You have someone say, hey Steve, I have some data, I need you to produce a report. Or are you still in the sort of vortex?
I can tell you more about the Hadoop version because we haven't gotten this one to that stage. But basically what the workflow for a biologist would be is that they run a program, they fill out a form which talks about the biology. It writes a file. You remember, you probably don't remember, but there was the last file I gave it was called input, gosh, I don't even remember what it was called. But essentially it's generated by that program. And what the biologist does is send that program to the standard biology program. That's the input for it. What I did with the Hadoop program is essentially I wrote something that took that file as the input and ran it and gave you back what the program would give you running on a single machine. So one of the things that I found that in both Spark and Hadoop I want to do this is that the last step involves a deliberate partition of one. Everything goes to one machine, it writes a file to HDFS, and that file is in the format that a single machine would generate because the next stage in everybody's pipeline wants to eat that. And that rarely takes a huge amount of time. These are not gigantic files when you get done.
Okay, I have a question. I have a question. Like when you were saying that you were facing problem while you were scaling up this, basically whenever data getting increased more than 400,000.
That's what I could do this morning.
Yeah, so
yeah.
more than 400,000. So what kind of problem you were facing? And and you mentioned about it's some kind of memory problem.
Yes.
Okay, so to me actually I'm not a biologist, I don't know like much. So to me it's a more like you have a data set A and you have a data set B. Both the data sets are larger data.
Yes.
So you are basically joining these two data sets and trying to get a common data set in between them.
Correct.
So in order to do that in the parallelized version, one of your data sets need to fit into a memory at least in one node.
You've already broken it up into much smaller divisions. And one of the tricks that I have used is if you find that you have a bunch of spectra and a bunch of peptides, they need to score against each other. You need to generate all pairs. Instead of scoring them in one function, I break them up. And in this case, what I will do is I will split the bin into multiple bins, each of which contains a subset of the pairs, and score that. the real question is, I need to identify when I need to split it and how many times I need to split it to keep things small enough. And I'm still working on that, on figuring out what those numbers are.
Whether you do two-step accumulation or the three-step accumulation, correct? Because
It's always two-step accumulation, because when you do the scoring, you're going to score all of the pairs that you want to score. How many partitions and bins you score them in is something that you get to decide. But you're going to get scores for every spectrum pair that you care about, and then you just have to sort them by spectrum and choose the best one.
so just to deal with the memory problem, like you told, you were running with a 15-node cluster, and each executor you have 14 gigs of memory.
Right.
And but essentially it's not the number of cores also there, because each core works on the particular ladder function or the unit of program you are in. So what is your number of cores for executors?
Number of what?
Number of cores.
Cores. Oh, I believe I'm not sure whether it's 4 or 8 cores per node.
That'd be cores per node. But I don't think we've covered how many executors you have across the whole cluster anyway.
Yeah, I'm not even sure about that.
That'd be cluster configuration that we'd have to dive into.
Right.
It's basically some Spark configuration, not the cluster configuration. Your cluster may be 15 node. I can say that I need 30 executors, and basically on top of 30 executors, I need suppose 10 cores. So each executor will be basically segmented into 10 cores. Means 10 parallel. 10 parallel.
Well, they're using Mesos for their resource allocation. So based on their total number of core resources across the whole cluster, they'll be able to grant each worker a set number of cores and a set number of memory. That'll just be a function.
Why I'm saying is that because sometimes this manipulation and balance between the cores and the executors sometimes actually help to resolve the memory problem. If I do not have the given that if I do not have right input format, then it actually cannot make it happen. Okay, that's all. Thank you.
You should talk to me offline.
Any other question?
Okay, thank you very much. Yeah, question?
What did you discover at the end? Not something Spark, but in terms of biology.
Wait, in terms of biology, what's been discovered?
Yeah.
Oh God, this is a technique. It's been used. The people in Norway, the biologists who are doing this, are in the Cancer Institute. So they would probably be looking at certain proteins that we're really interested in terms of cancer cells and how they replicate. But there's a lot of other problems that this is relevant to. It's a tool.
Thanks, Steve.